You are A. If you are A, you have antigen A on your red cell, and that's why you are A. If you are B, forget about the positive or negative. You have B antigen on your red cell. Don't distract the lecture. If you are AB, you have AB antigen, A and B antigen on your red cell. And if you are O, you lack all the antigens. You don't have A, you don't have B, you don't have AB. But if you are positive, that means you have resource antigen along with whatever you have, whether it's A, whether it's B, whether it's AB, whether it's O. But if you are resource negative, meaning you lack the resource antigen. You know about Landsteiner law. So what's Landsteiner law? Landsteiner law states that if you have a particular antigen on your red cell, the corresponding antibody will be absent. Will be absent. It's the same thing we are saying. It will be absent in the plasma of the person. If you have a particular antigen, so on your red cell, that's what the law states. So meaning that if you have A antigen on your red cell, what's the corresponding antibody to A? No, it's anti-A now. Antibody to A. Antibody to A will be absent. Why must the corresponding antibody be absent? That's natural phenomenon. Okay, why must it be absent? Otherwise, the antibody will be reacting with the antigen. That's what the law says. So meaning that if you are resource positive, the corresponding antibody, what's the corresponding antibody? Anti-resource will be absent in your plasma. But if you lack resource antigen, then you have what? You have the antibody. You have resource antibody. Why is it like that? Like I've just told you, why must you have a corresponding antibody absent when you have a particular antigen? Why? So that there will be no agglutination. Antigen and antibody will react with each other, and if you have agglutination in your blood, what's the implication? It will be destroying the blood. It will be destroyed, and that's what happens with resource hemolytic disease of the liver. So what's the lifespan of red cell? So if you have so much amount of antibody, corresponding antibody, so the lifespan will be reduced to like 20 days, 30 days. And what's the implication of that? There will be anemia because the red cell will be destroyed prematurely, and then you have anemia. So what's the response to that? Is called hemolytic anemia. What's the response? How will body respond to that? It will try to produce more red cell. So you now start seeing mature red cell. So how will you determine that the body is responding to the anemia of hemolytic anemia? What test will you do? Reticulocyte count. You see retic will go up. Retic, you know about reticulocytes. Reticulocytes are immature. By the time you start seeing immature red cell in the film, and then the patient is anemic, and you start thinking about hemolytic anemia. So these antigens, red cell antigens, they are proteins attached to the red cell. They could be glycoprotein or glycolipids. We know the important ones. What are the important red cell, common ones that you must do before you do transfusion? the important red cell antigen that you must check before you transfuse blood, because the purpose of this is you want to transfuse, somebody is bleeding, somebody needs blood, you must give a group compatible, and that's why you must check the antigen on the red cell, and that's why you must do the blood group. And that's why some of us, when we are carrying our ID card, we are advised to indicate our blood group in case you have road traffic accident. We'll just check from your ID card and then quickly go and get blood of the same group. So why must we, what are the important antigens, the red cell antigens that we must know? That if you don't check, your patients or your clients will have problem. ABO results, those are the important ones. ABO results, ABO results, and AB. So who knows of any other red cell antigen? There are 400 or more. Yeah, 400. Yeah, can you just sit down and just tell me? You have the kid, you have the Kel, you have Louis, you have Duffy, you have Duffy. Continue now. We are going to 400. You have big N, big N, big S, small s, big I, small I, cartwright, SG. That's all I know. Okay, so there are many types, but not all of them are of clinical importance. Okay, they must be able to cause hemolytic transfusion reaction or hemolytic disease of the newborn for them to be clinically important. What do I mean by hemolytic transfusion reaction? So you have given an incompatible blood, somebody needs blood, PCV before transfusion was like 17%, and you have given 3 pints. So after your 3 pints, PCV came to 13. Yes, because if you have given a wrong blood, the antibody in the recipient will be destroying the red cell of the blood you have given. And that is what is called hemolytic transfusion, hemolytic transfusion, hemolytic transfusion. So that is why it is important for you. So it is important for you to determine the blood group before. So let me just put on the okay. So like you have antigens, ABO antigens, you also have antibodies. Okay, so you have naturally occurring antibodies and then the immune, and so those are the two major types of antibodies that you have. The naturally occurring antibody are usually IG. You know about the GADM, G antibody GAD. So the naturally occurring antibodies are IgM, and they are anti-A or anti-B. Okay, anti-A is for A antigen and anti-B is for B antigen. So they are called naturally occurring because they are IgM and they react maximally at cold. So they are called cold antibodies. Unlike the immune antibodies are IgG, and they react at warm temperature. The resus antibodies are immune antibodies. They are IgG because they will be able to cause the placenta. Okay, so like you have the naturally occurring, which are IgM, the anti-A, and then the anti-B. You also have the immune antibodies. Okay, so it is now known that there are two types of A antigens now. So you can be A1 or A2, but for your level, just know that there is A antigen. So if you have A antigens, you have A antigens on red cell, on your red cell. But A antigens can be, it is not of clinical importance whether you are A1 or A2, okay? But you are group A, okay? And I want to mention again another important antigen called H antigen. H like in hippopotamus, okay? H is the precursor antigen that the A and the B and the O are made from. The H antigens are the precursor antigens. So if you have H antigen, so what will be absent in the plasma of the individual? Antibody to H. Okay. So H antigen is the precursor from which all the antigens are produced from. Okay, so but we know some individual lack their they lack H antigen. They lack H antigen. So if they lack H antigen, meaning that they will have H, they have H antibodies. Okay, so those that lack H antibodies are considered as those that lack H antigen but have H antibody are considered to have the Bombay phenotype of the O. Okay, they are O because the H antigen is highest with O, followed by A2, followed by A2B, followed by B and A2 and A2B in that order. So H antigen is the precursor from. So if you have if you lack H antigen, that means you have H antibody following the Lansdowne law. If you lack H antigen, you lack H. I hope you are following me. Okay, so the individuals that lack H antigen but have H antibodies are called the Bombay phenotype. Bombay is from which country? Okay, Bombay is from which? So they are. Oh, so this is the. You can see the frequency distribution of blood groups. Which one has the highest among Nigerians? The majority of Nigerians, 52% of Nigerians are O positive or O generally, followed by B, 23%, followed by A. AB is rare, okay? AB, so just 4% of Nigerians are B. You can see the frequency for British, for Indians, for South American Indians. Where is South American India? Where are they from? South American India. They are from Brazil, Argentina, Cuba, Chile. They are referred to as South American Indians. You can see they are predominantly O. What about Australian Aborigines? Where are they from? Who are they? I know they are Australian. Who can describe the Aborigines? They are natives. They are the black Australians. So you can use blood group to determine demography. You can use it for paternity testing. You can use it for forensic medicine. So for example, if you claim to be South American India and then you are A positive, this is assuming there is no intermarriages, typical because during intermarriage it's possible to have a change in your blood group. Or if you claim to be Australian Aborigines and then you are group A, okay, so they will know you are lying. Okay, so you can use blood group for paternity testing in some cases. I will explain how it can be used for paternity testing. Then if somebody is involved in a murder and then they took sample of the incidents, and then the suspect is AB and the sample that they took is AB, while the patient or the corpse is O, you know, you can use that to say, this, although it's not too reliable like DNA, but DNA will be more reliable. So they use it for anthropology, for forensic medicine, paternity testing. And that's why you have the frequency. This, but in Nigeria it will be difficult because cut across. Nigerians have all the various type. So it will be difficult to say it is, you are not a Nigerian, because we have AB, you have B, because of maybe intermarriages among Yoruba, Igbos, and the Hausas. Okay? So, like I was telling you, there are major type of antibodies. You have the naturally occurring that are IgM, and they react maximally at cold, 4 degrees. And then you have the immune that are IgG and react at 37. Okay, so if you want to give blood, you know about the phenomenon of universal donor and universal recipient. So which one is the universal, which of them is the universal donor? Okay, what about the universal recipient? AB. So O can give blood to any of them as long as the resource is compatible, whether A or B. But there's what is now known as dangerous universal donor. And that is why it is advocated that you give a group compatible, group specific blood. What is the meaning of giving a group specific blood? A to A, B to B, AB to AB, O to O. The reason is that because of the idea that O is universal donor, they give O to A, they give O to B. So individual with O that needs blood, they don't get to get O. So it creates scarcity of O. Another problem about this dangerous universal donor, like I said, there are 2 types of antibodies that you are carrying. You know, you have O, meaning that you lack which antigen? You lack A antigen and B antigen. Okay, and then you have H antigen. Okay, but you have which antibodies? You have antibody A and antibody B. Okay, but if you have H antigen, you lack H antibody. Okay, so if the antibodies, this anti-A that are expectedly to be naturally occurring IgM, because some of them also could be immune antibody. They could be anti-A, IgG anti-A, IgG anti-B, but it is expected that they should be IgM anti-A. If you are group O, you have the antibodies, IgM, and I hope I'm on the same page with you. Naturally occurring antibody reacts at 4 degrees. Your body temperature is not 4 degrees, and that's why it is so that there will be no reaction. The immune antibody will react at 37, and your body temperature is 37. Okay, and immune antibodies are IgG. Okay, naturally occurring antibodies are IgM. So if you are group O, it is expected that you have naturally occurring anti-A and anti-B. But we now know some individuals have immune anti-A and immune anti-B, which are what type of antibody? IgG. Immune antibodies are IgG. At what temperature do they react best? Okay, so you've now given your O. The donor has high percentage of immune antibody, anti-A, anti-B, to individual that is A. What do you think will happen to the antibody antigen, anti-A in the donor that is immune, and antibody, antigen A? What do you think will happen? And don't forget it's 37, not cold. What do you think? There will be agglutination. And what will happen? There will be destruction of the rest. I hope I'm on the same level with you. Am I on the same level with you? Okay, I will say it again. I said there are two types of antibodies. Okay? One is called naturally occurring antibody. Okay? And this naturally occurring antibody, that is why it is natural, it can live in you, reacts best as 4 degrees, and usually IgM. You also have immune antibodies that are usually IgG and react at 37 degrees. Okay? So, and I said if you are group O, you lack antigen A and antigen B. Don't forget your Lansana law. That is, if you lack antigen A and antigen B, what will you have in your plasma? Antibody A and antibody B. Okay? So, and you now have a high percentage of this immune antibody, more than expected, more than the naturally occurring. Okay? And you are O. And then you now donate your blood, that O blood that has a high percentage of IgG anti-A and IgG and anti-B that reacts well at 37. You give it to A or B individual, okay, in the name of universal donor, or you give it to AB individual in the name. So what would now happen? The antibodies that are immune in the donor, the anti-A, if you have given it to A blood group individual, will react with the A antigen. Is that difficult to understand? If you have given it to B individual, the anti-B in the O that is immune will react. And what's the implication of that? It will cause hemolysis. So it is possible to give blood pre-transfusion 17, 3 pints, post-transfusion 13 PCV. Because if you have given the wrong type, that is why it is advocated that you give a group specific blood rather than group compatible. Because if you do a cross match, it may be compatible. If you do O cross match with A, or cross match with B, or cross match with AB, it may be compatible, but you are not assessing the immune antibody content of that O. So it is possible you give that blood and there will be hemolysis. And that is why it is now recommended that you give group specific rather than group compatible. Do we know the implication of this dangerous universal donor phenomenon? I'm going to the mosque. I'll come back. I'm not in a hurry. Okay. When are you after the meeting? So that is what is. Who can explain what I've just said about dangerous universal donor? Not you again. I want to be sure it is understood. Ogbo, they are pointing at you. Yeah. You don't understand. You don't understand. Wow. This is the third time I'm repeating it. Oh. Who understands? So majority do not still understand. No, not you. Majority. So I should repeat it again. Okay, I will repeat it again. So, but this time it will be interactive to be sure that you understand. So if you are group O and I'm group B, then it is assumed that you are universal donor. You want to give me your blood, I can take your blood. Is that not the what you are taught? Okay. So I want to tell you that that one could be dangerous. And I told you that there are two types. Okay, if you are group O, which antibody do you have in your plasma? Antibody A and antibody B. And what are the two types of this antibody? The naturally occurring are reacting best at and they are IgM. The immune antibodies react best at and they are okay. So you are the donor, you are the group O and I'm group B, and you have high percentage of immune antibody, the anti-A and anti-B in your plasma. Okay, more than the naturally occurring, because if it was naturally occurring there will be no reaction because 4 degrees and my body is not fish. Temperature is like 4 degrees, human is 37, so there will be no problem. Okay, but if you now have a high percentage of immune antibodies, the anti-B, that is IgG, you are going to give me your blood and be positive. What antigens do I have in my blood? What antigen do I have in my blood? Antigen B. So, and you are giving me blood that contains anti-B, that is IgG, that is best reactive at 37. My body is 37. What do you think the plasma in the O will do to my red cell? That will be agglutination. But if it was predominantly naturally occurring anti-B, there will be no agglutination, because despite the fact that it has anti-B and I have B antigen, but because it's naturally occurring, it's IgM, there will be no reaction because it reacts best at 4 degrees. My body temperature is 37. So you are going to explain to me what I've just said. Yes. Dangerous universal donor phenomenon. Write an essay on dangerous universal donor phenomenon. Number 1, compulsory. Explain what I've just said. Or volunteer. Who wants to volunteer? Or you still don't understand? They are workouts if you don't understand. Explain now. Explain if you understand. Explain. Don't waste time. They will soon switch off the gen. Who wants to volunteer? Okay, volunteer. Let's listen to him.
The reason that it's called the dangerous blood
Universal donor.
dangerous universal donor is that the universal donor, that blood sample may contain a large amount of antibody and an antibody for body A. So when you introduce it to a patient, because the body temperature for those antibodies is 7 degrees, and there's a large
You
amount
must mention that they are immune. They must be immune, because if you give naturally occurring, it will not react. So if you have a hyper, I'm sure you will understand before I move on. Okay, what about this Bombay phenotype? I mentioned Bombay. Who understands it? Hmm? The Bombay lack H antigen, but they have the antibody, they are O. Okay, so meaning that all antigens, all blood group also have H antigen. Generally, if you are O, you have H because it's the precursor. It's the precursor of the A, the B, so it is used up, and that is why it is highest. If you see it is highest in, look at it. It is highest in O, followed by A2, followed by A2B and B. So despite the fact that you are O, you still have H. Have you heard about H before now? No, this is hematology, this is not physiology. Despite the fact that you are O blood group, you still have H antigen. H like in hippopotamus. So because if you have H antigen, you will lack H antibody, according to Landsteiner. Is that correct? So they now discovered some individuals that are O but lack H antigen. Okay, they are called the Bombay phenotype. Because they lack the H antigen, they will have what in their plasma? H antibodies. Okay, so if you say because it is O, you want to give O, and the person is the Bombay phenotype, what's the implication of that? The H antibody that the Bombay individual has will be reacting with the H antigen in the red cell of the donor. Does that make sense? So what will be the implication? Hemolytic transfusion reaction. So what must you give them Bombay phenotype individual? Bombay. You must give them Bombay phenotype blood. You must give them Bombay phenotype blood. We are not in India. You are not likely to come across Bombay phenotype here, except you are working in Indian hospitals in Lagos, or you, after your graduation, go to Saudi Arabia to work, Dubai to work. There are plenty of Indians, and that's why you must understand this Bombay. Okay, so I think those are the two. So if you want to do a blood grouping, there's what is called forward grouping and reverse grouping. Have you heard about that before? You have not heard about forward grouping and reverse? Eh? Okay. Okay, forward grouping. I hope you are listening. Okay. Forward grouping. So if you want to do a blood grouping, okay, you use the anti-sera. You've heard about anti-sera A and anti-sera B. Have you heard about anti-sera A and anti-sera A has antibody A? Okay. So but anti-sera B has antibody B. So if you now mix the red cell, if you mix my red cell, if you want to determine my blood group, okay, you want to determine my blood group, you now mix my, so you put your anti-sera A, anti-sera B. Okay, let's forget about results for now. You put anti-sera A and anti-sera B on a tile, and you put my blood, and there is agglutination in that of anti-sera A. What blood group am I? Think again. Eh? Eh? Listen. I have anti-sera A on the white ties, antisera B on the white ties. I don't know my blood group, but I want to determine my blood group. So I now draw blood into a syringe and put a drop into the antisera A, put a drop into antisera B, and there is agglutination in antisera A. What blood group am I? so so think again. You put antisera A, you put antisera B, you don't know my blood group, okay, and there is agglutination, antibody, the antisera A. For you to have agglutination, that means I have antigen A. But if there is no agglutination, that means I am, how can it be O? O has no antigen. O has no antigen. Okay, so antisera A has antibody A. Antisera B has antibody B. So and I want to determine my blood group. For you to have agglutination, you must have an antigen that corresponds to the antibody. So you want to determine my blood group. I don't know my blood group, but I have added my blood containing red cell into the antisera A. And antisera A, for it to react with my red cell antigen, meaning that I must have antigen A. And if I have antigen A, what blood group am I? A. That is a forward blood grouping. What about the reverse blood grouping? This will turn your brain a little bit. Okay. So I want to know whether I don't know my blood group, okay? And I have known blood group, known red cell A, and known red cell B, okay? I now mix a serum. You know a serum, okay? You mix it with the known A. I hope you are listening. Eh? Okay, let me explain again. Let me explain. So how do we get serum? Where do we get serum from? You can get plasma, you can get serum from the whole blood, okay? And serum or plasma is supposedly containing, is it the antigen and the antibody? Antibodies. Okay. So I don't know my blood group, but you know I have my serum. I've given you my serum. I hope you are following. I don't know what it contains, whether it is anti-A or anti-B. But I have known red cell, known A, and known red cell containing B antigen. Okay? Known A, I've washed it, I've grouped it, and I know this is A. I've labeled it A red cell antigen, B red cell antigen. But my serum, I want to use my serum to determine my blood group. So I now mix the red cell A, known A, with my serum, and there is agglutination, meaning that there is anti-A in that serum. So if you have anti-A in your serum, what blood group are you? Exactly. That's B now. That's a reverse grouping. Reverse grouping. So who is going to explain forward and reverse grouping to me now? Okay. Maybe one. For forward grouping, you need antisera. I want to explain myself again. You need antisera, okay? What do they contain? antibodies, okay? For forward grouping, you need anti-sera, and then you have the red cell of the individual, but you don't know the group of the person, okay? So how do you do that? You spot the anti-sera, anti-A and anti-B on a tile, and then you mix what? You mix the red cell, you mix the red cell containing the antigen, okay? You want to determine the, and there's agglutination with anti-sera A. For you to have, what group is that? If there's, what group do I belong? I'm A. Because for you to have agglutination, meaning that my red cell contain antigen A. But if there is no agglutination, what group do I belong to? I hope it's clear. I want somebody to explain the reverse, okay, reverse grouping. In reverse grouping. Okay, listen, please listen, those at the back. Reverse grouping. And you don't know the group. Group of the person. Okay, but you have the serum. Okay. Known. Known A and known B. Mix.
So if there is agglutination of the serum with the A, that means the person is. Because,
why?
Because normally the body cannot have antibodies against it. Like if there is an antigen in the body, it cannot have that corresponding antibody in the plasma. So that means if you are mixing with it, for you there to be a reaction.
Which one are you talking about? Is it the A known?
The A.
Known A, okay. The known A. I hope you are following.
Yes, sir.
If you mix the known A with the serum, unidentified serum, okay, and there is agglutination, that means the individual is Y. Because the anti-serum must contain anti-B to cause agglutination. Okay. So if there is no agglutination, I hope somebody has understood here.
For the natural and
Naturally occurring.
is there a way to know which one is like highly present?
Yes, now that one is postgraduate. When you come back for masters in hematology, you can determine whether the individual has the quantity of the naturally occurring anti-A or anti-B. You can determine that. Whether it's immune, you can determine that. I think we have not started the results at all. So this is just about all the, okay, we'll not talk about results. Okay, results was, okay, ABO was discovered maybe 1900. We are in 2023. That's 123 years. The A and the B and the O. But AB was discovered by the student of Landsteiner shortly after that. But results was discovered in 1939 by Levin and Stentins. How did these people discover results? Okay, I hope you are listening. Yes. Okay, they injected rabbits with the blood of a rhesus monkey. I hope you are following. I want you to listen. Those are the back. So they now harvested the serum. What will the rabbit contain? The serum, the rabbit, what will it contain? If you have injected rhesus blood into rabbits, rhesus monkey's blood, so the rabbits will produce antibodies to rhesus monkey's blood. Is that clear?
Yes.
Okay, it's not clear. Okay, I said this is a rabbit, this is rhesus monkey. There is a monkey called rhesus. I draw the blood of rhesus monkey, the red cell, inject it into the rabbit. So naturally, the rabbit will produce antibodies against that rhesus monkey wrestle. Does that make sense? Body will try to fight with antibody against corresponding antibody. Okay, they now withdrew the serum of the rabbit containing what? Antibody to rhesus, and now started mixing European blood with that serum. European men and women. This is not in vivo, it's in vitro. So don't be afraid though. It's in vitro. And they now discover that 85% of Europeans agglutinate to that serum. The serum of the rabbit containing anti-rhesus agglutinates the red cell of human, 85% of human. So they now concluded for it to agglutinate, those people must have rhesus antigen. That's how they came about it. So 15% did not agglutinate, meaning if you do not agglutinate to the serum supposedly containing rhesus antibody, that means you lack, so you are rhesus negative. That's how it came about. You are rhesus negative, meaning that you lack rhesus antigen, and then it is expected that you have rhesus antibody. But because it is immune, you are not likely to have, except there has been fetal maternal hemorrhages. And that's where it comes on, that if you are rhesus negative as a woman, and during delivery, as you are trying to cut the placenta, there is a mixture, and you are carrying a rhesus positive baby. The father is rhesus positive, okay, and the baby is rhesus positive, okay, and as they are trying to cut the placenta, the blood of the baby mixes with that of the mother. Because it is immune, it's IgG, so the mother will now develop antibodies to rhesus. Because naturally, if it was IgM, there will be no problem. So if you now have antibodies to rhesus as a woman, and then your subsequent pregnancy, you are also carrying a rhesus positive baby, but you have acquired antibody from the previous pregnancy, and because it is IgG, I keep repeating, this immune, because if it was IgM, there will be no problem. The IgG anti-rhesus antibody that is being carried by the mother can cross the placenta during pregnancy into the baby that is rhesus positive. And what will happen if you have a rhesus positive and rhesus negative antibody to rhesus? There will be, so depending on the quantity of the antibody that the mother is carrying. Hello, Dr. Odubono. I'm in class. I will see you after the class. I'm in class. Yeah. So depending on the quantity of sensitization, that's the right word, of the mother, will determine whether the baby will live till birth or not. If the quantity, we can also quantitate as hematologist if you come for postgraduate. If somebody is rhesus, a woman is rhesus negative, and there is previous history of intrauterine death or death after delivery, and the husband is rhesus positive, we will collect the serum of the mother and quantitate the amount of rhesus antibody in the mother. Then we will advise the obstetrician, this titer is very high, you must terminate this pregnancy. If you don't terminate it, this baby will die in utero. There's the technology now to do that. We can do that here too. So you can quantitate the degree of sensitization, but some degree of sensitization is just very little that will not harm the baby. Okay, so you can determine that. So that's the importance of resource, antigen, and antibody.
You already have the program. Why do you have to terminate?
Okay, that's a good question. Since we have rogam, why do you have to terminate? Rogam is an anti-resource. Rogam is the antibody that is meant to destroy anti-resource. Okay, so depending, we give rogam during pregnancy. Depending on the quantity of the anti-resource that the woman is carrying, we can advise obstetrician give one shot of rogam before delivery. But if the quantity of the titer, the anti-resource is not high, we just advise just monitor the baby in utero with ultrasound, make sure that the baby is doing well, and then immediately after delivery, maximum 72 hours, you need to wipe out the anti-resource antibody in the mother with another antibody that is meant to destroy that anti-resource, and that is rogam. So depending on, like I said, I keep saying, depending on the titer, it may be beyond what rogam can handle. Okay, so depending on the titer, it may be beyond what rogam can handle. So we have a clear understanding of the, but it's much more than that. They now discover that the anti-D or the D, the result is known as the D antigen. Is D antigen. I hope you are aware. Result D positive, result D negative. Okay, but they now discover there are more than another letter involved in results. So you have the big C, the small c, the big E, the small e. There is a reason for that. I will explain. I will explain because if you are results positive as a woman and you are married to results positive man, it is possible for you to have a results negative baby. They shouldn't accuse you of infidelity. Are you surprised?
Yes.
You don't know it's possible. And if you don't consider this big C, big E into your look, into your perspective, then, okay, let me explain with this one. So this is the haplotype of an individual. Haplotype meaning that you have inherited one from your father and one from your mom. So if you do your haplotype, you have the facility, you are going to be any of these. So this is the abbreviation. It's called the Wiener nomenclature. This is a Fisher nomenclature. Okay? So your results D, for you to be results positive, you must have one big D at least in your haplotype. I hope you are following me. Listen very well. For you to be results positive, you must have one big D. Absence of big D is known as small d, and meaning that you are results negative. This is hematology and not physiology. Okay? Absence of big D. If you lack big D, for this lady is results negative. Abi? So meaning that you have small, you don't have big D. Because if you have big D in your haplotype, So this is likely to be your haplotype. Is there big D here? So it's small, so you are rhesus negative. Okay. Imagine now, I hope listen well. This is the haplotype. This is the haplotype of the husband. Okay? And the same haplotype for the wife. Okay? And the husband has donated this to the baby, and the wife has also donated this to the baby. Where will the baby get big D from? Where will the baby get the big D from? Eh? No, I said, it has to be one per person. This is the haplotype of the person. You have to donate one of this to your baby during fertilization. So if the father has donated this, the same haplotype, the father and the mother, the same haplotype. The father has donated small d, small c, and small e. And the wife has also donated small c, small d, small c, and small e. Is there big D in the baby? So the baby will be rhesus negative, despite the fact that the mother is rhesus positive, because the mother has one big D, and the father is also rhesus positive, has one big D. Like I said, if you have a big D, you are going to be rhesus positive, except the degree of positivity. If you have, like this one, this big D here, and there is a big D here, will you come? This person will also be rhesus positive. This person will be rhesus positive, but the number of D antigenic sites is more in which one? The second one. Okay? The number of D is more in the second one. Does that make sense? Can you explain why it is possible for you to have a rhesus negative baby, despite the fact that the father is rhesus positive and the mother is rhesus positive? Who wants to volunteer? Father is rhesus positive, mother is rhesus positive, but baby is rhesus negative. Who wants to try? Okay, no, I don't. You understand it very well. I want another person. Okay, try. Let's have one class. I want to put on the slide so that it will be clearer to you. Use that to explain why it is possible for you to have a rhesus negative baby, despite the fact that the mother is rhesus positive and the father is rhesus positive. Why?
Okay, so both parents have two haplotypes, and if they both have big D and small d during fertilization, it's possible that they both give small d to the child, and small d means vicious negative. So if they both give small d to the child, there's no way to get any.
Simple. So so this is a abbreviation. This is the abbreviation. It's called winner. It's big R and small. So this is the common rhesus genotype. This is the UK haplotype. In terms of frequency, 31%. When I was in US, I wanted to do my haplotype, but they couldn't get my vein. So I couldn't do it. So I would have done my haplotype and know whether I'm big R and small r, or big R or big R. Okay? So this is okay. There is another phenomenon called weak D. Weak D, weak D, weak D, weak, weak, weak D, weak, weak, weak. WEAK. I hope you are enjoying yourself. So, like I said, the antigenic site varies from one person to the other. Okay? Like some people have two big D, some have one big D, and yet they are still results positive. So the individual with fewer D antigenic sites are known as weak D or DU. Weak D. Okay? The clinical implication, the clinical implication of that is that the individual, because it's going to take a longer, if you mix your anti-D with the blood of the person, okay, because they don't have many antigens, if you don't wait long enough, you will label the person as results negative. You will label the person as results negative because ordinarily it should take 1 minute for the agglutination to come up. But if you don't have, if you have weak D or what is called DU, it's going to take a longer period. So what's the implication of being labeled as weak D? What's the implication? Yeah, we will waste the patient's money to buy Rogam. You'll be wasting the patient money to buy Rogam after delivery, whereas the person is D positive and not D negative. Okay? Does that make sense? Then if you are giving blood, you will now start looking for results negative blood. You will label the person as results negative. You start looking for results negative blood. Okay? So what's the implication of that? If you give results negative to weak D, there will be no, no problem about it because the D antigen itself is weak. It's weak. So it will not cause any reaction except that you will be wasting results negative blood that is scarce. Do you understand? You will be wasting results negative blood. So that's about weak D. Weak D, weak D or DU. 8% of Nigerians, 8% of Nigerians have weak D. Maybe you are weak D. You understand? So you need to see hematologist and you have been labeled as results negative. So if you are weak D, your husband will just be wasting money to buy Rogam. Eh? They will label you as results negative. After delivery, you will ask your husband to go and buy Rogam. Rogam is like maybe 70,000 now. Yes. If you are still looking for money to pay hospital bills, you will be buying Rogam. Do you understand the phenomenon of weak D? So 8% of Nigerians are weak Ds. They are DU. 8. 8%. So I will just round up now with Coombs test. So there is okay before that. So there is Duffy, as you have said, there is Duffy red cell antigen. They can all cause, these antigens can cause hemolytic transfusion reaction. So apart from the ABO results, there are other red cell antigens Even if you give group specific that can cause that we don't check for. We don't check for Duffy, we don't check for Kid, we don't check for Kel. They can all cause hemolytic transfusion reaction. But if we give a group specific blood and there is evidence of hemolytic transfusion reaction, then they will call hematologists. Hematologists will do antibody screening and identification in the recipient. Okay, so we'll check whether, because we don't do apart from A, B, O that we do, we know there are other antigens in the red cell that can cause hemolytic. So we'll identify the antibody that is present in the recipient. And how do we resolve it? You give red cell that lack the antigen. Does that make sense? Does it make okay? Okay, you have the person is A, and then you have given A blood. Okay, that is a group specific. You don't expect hemolytic transfusion reaction, because if you are giving A blood, the individual has anti-B, the donor has anti-B, okay, and the recipient does not have B antigen, so there should be no. But we know, and then you have given results compatible. Person is results positive, you are giving results positive. But we still know that some antigens in the red cell that we don't check for routinely can also cause hemolytic transfusion reaction. And then instead of having a rise in the PCV, there is no rise, and the individual, the recipient is jaundice, meaning that the individual is hemolyzing. What is responsible for jaundice in hemolysis? Breakdown of red blood. What is the component? The bilirubin. From where? From hemoglobin. So we know that. So what we do is to do antibody screening of this, some of these antibodies, and like the Duffy antibody in the recipient. You can do Duffy, you can do KIT, you can do MNS. They can all cause, and then identify red cell that lack this, because if you have red cell that lacks this and the recipient has this antibody, there will be no do you understand? And that is why it is not common to have this, but it is possible. And this can all cause hemolytic. The big M, the big N, the big S, and the small s, they are all red cell antigens you have not heard about before today. Have you heard about them before today? So I'm just telling you that. So this is the kid. Again, 70% of blacks are negative for Duffy. They are negative, they lack Duffy antigen. If you are black, like you and I, 70% of us, Plasmodium viva requires Duffy antigen to penetrate the cell of your red cell and cause parasitemia. So if you lack Duffy antigen, will you be infected with Plasmodium viva? Eh? You won't, because that is why it is rare. This is MCQ, part of MCQ question. Plasmodium viva is rare in West Africa because of what? Because we are negative to Duffy, but it's possible because it's not all blacks that are negative. Okay, so that's the importance of Duffy, and then P viva. So if you lack Duffy antigen, you have what in your plasma? Duffy antibody. Okay, so we'll talk about Coombs test and round up. Okay, so there's SG. Don't forget, you read this SG. Is found on X chromosome. It's also a type of red cell antigen. So there are two types of Coombs tests. Okay, do you know have you heard about Coombs test or direct antiglobin test? Yeah. Direct antiglobin test. Have you heard about it before now? Yeah. Who wants to explain? You want to explain? You don't have idea. So there are two types. There is direct antiglobin test and there is indirect. Just like you have forward grouping and you have reverse grouping. You can also have direct antiglobin test or direct Coombs test. Why do we need direct Coombs test? Okay, mother is rhesus negative, baby is rhesus positive, father is rhesus positive, there is hemolysis. You want to check in the baby's red cell whether it has anti-rhesus, and that will confirm why there is hemolysis. Because if there is no anti-rhesus antibody on the red cell, there will be no reaction, there will be no destruction of the baby's red cell. Does that make sense? Mother is, let me explain myself again. Mother is rhesus negative, baby is rhesus positive, and after delivery, father is rhesus positive after delivery. You now collect the baby's blood, red cell. You want to find out, is there rhesus antibody? No, we want to find out whether there is rhesus antibody on the red cell of the baby acquired from the mother. Is that difficult to understand? After delivery, you just collect the blood of the baby. That's what this test, direct Coombs test, is about. You know you can do that. You have never thought of it. Eh? That you just need, this is in vivo, it's not in vitro, it's not, baby is out already. You now collect 2 ml of the baby's blood. You want to determine, is there antibody to rhesus? Probably acquired from the mother. Because you know, mother would have antibody, especially if the mother has been sensitized anyway. So you want to confirm that this is what is causing the hemolysis. That is what Coombs test is all about. Direct. Then indirect Coombs test, you want to determine, is used in cross match, whether you have antibody. I want to be giving blood by the HOC Demilade. Demilade is donor, I'm the recipient. I want to determine, that's the indirect now I'm explaining, whether I have antibodies to any of the Demilade's red cell antigen. If I have antibodies to any of the Demilade's red cell antigen, that means if they give me Demilade's blood, my antibody will what? Destroy. That's indirect. That's indirect because you are assessing the presence of antibody in the serum of the recipient as indirect. But for direct, you want to determine whether there's an antibody on the red cell. Okay, so how do you determine whether there's antibody? So I've given a scenario. Why do you want to determine whether there's an antibody? Why? On the red cell of the baby. Why? Because particularly if you have a history that the mother is rhesus negative, the father is rhesus positive. Well, no, I want to digress a little bit. Mother is rhesus negative, father is rhesus positive, baby is rhesus negative. Is there a need for Rogam? Why? If the baby is rhesus negative, there will be no need for Rogam because the baby does not even have the rhesus antigen anyway to destroy his blood or her blood. So Rogam should not be given routinely for all rhesus negative mothers. You should check first. You know, it's possible, like I've said, it's possible for you to have a mother that is rhesus positive or rhesus negative, and the father is rhesus positive and the baby is rhesus negative. You have explained that it is possible for you to have a mother who is rhesus negative, not with D, not true rhesus negative, and the father is rhesus positive, depending on the haplotype you are talking about. If the mother is rhesus negative, lacks big D, so has small c, small d, small, or big C, small d, small e, or small c, small d, no big D, has donated her haplotype, one that lacks big D, and the father, the haplotype, has one big D and one small d, and has also donated that of small d. Okay, so the baby cannot get big D from anywhere, so the baby will be rhesus negative, so there is no need. So what we do in direct Coombs test, which is meant to be for rhesus positive babies carried by rhesus negative mothers, okay, is to get the blood, okay, and wash the blood. What do you wash the blood with? Omo, jig, or what? No, you use normal saline. We use normal saline in the lab. We use normal saline in the lab to wash. Why are we washing the blood? Why? You want to wash off all antibody in the plasma. You wash and decant 3 times. Because if there is a corresponding antibody on the red cell antigen, if you wash 100 times, you remember the FAB, FC region of antibody is covalently bound. It will not be washed off. Okay? So you wash 3 times with normal saline. With what? Normal saline. Get 2 ml of blood from the baby and you wash 3 times with normal saline. The whole blood. You put whole blood in a test tube, put normal saline, centrifuge. When you centrifuge, all the unbound antibody in the plasma will spin, and then when you decant, they are going to decant with the sample. You will have a red cell at the bottom. Add normal saline again, all unbound antibodies, whether it's floating in the red cell, is not covalently bound, decant again. So what you have is a plain red cell, but if you have a corresponding anti-rhesus antibody on those red cells, if you wash 100 times, decant, wash, except you want to decant the red cell, it won't be washed off because of the FAB region and the FC region. The antigen antibody attachment is covalent. So after washing 3 times, wash 3 times, okay? What next? How do you now determine whether you have a corresponding anti-rhesus antibody on those red cells? How? How do you determine that? You add anti-human globulin to the cell suspension. You get a little bit of washed red cell. You now add your AHG, or it's called Coombs reagent. It was discovered by, what is anti-human globulin? It is an antibody to your rhesus antibody. Antibody against the bound rhesus antibody. Okay? Antibody against the bound rhesus antibody. Somebody will explain. Okay, I hope you are following. So because you have an antibody against the antibody, so it will now form a clump. It will form a clump. If there was an antibody on the red cell antigen, there will be agglutination. But if there was no antibody, rhesus antibody on the red cell antigen, the rhesus antigen, If you add an antibody, antibody will not bind antigen. It will only bind antibody. So if you add your anti-human globulin to the suspension and you look at it macroscopically or look at it under the microscope and there is no agglutination, you will conclude that that boy or that girl lacks rhesus antibody. The cause of the hemolysis cannot be rhesus hemolytic. It's either sepsis, drugs, or whatever. But if there is agglutination, what will cause the agglutination? I want somebody to explain. What will cause the agglutination? If there is what? If there is on of the baby, because all of the antibodies have been washed off during your washing process if they are not bound. So that is a direct Coombs test. That's a direct hello, good morning. I'm in a lecture. I'm giving a lecture now. Can you call me later? Okay. So that is a direct Coombs test. That's okay. Why do we need indirect Coombs test? I've explained why we need indirect Coombs test. Why do we need indirect Coombs test? Why do we need indirect Coombs test? You want to determine
If the recipients have the antibodies,
if the recipient has
antibodies,
antibodies that will react with the donor
antigen.
red cell antigen. So how do you do that? You get the serum, you get the blood. How do we get the blood? I want to give you blood. I want to give you my blood. Okay? And I've checked the blood group. You are group B, I'm group B. But I'm still scared that I didn't check for Duffy. I didn't check for Kid. And they can all cause hemolytic transfusion reaction. So I want to be sure that I don't have antibodies that will react with any of the red cell antigen. So what do I need to check? So I will collect the recipient blood, let it clot, get the serum, okay? Get the serum supposedly containing antibodies to the donor red cell, okay? So I will now mix the donor red cell with the serum. Hello, Prof. Mo wà ní class. Màá pè yín padà, mo wà ní class. So I will now mix the donor with the. I'll wash the donor red cell again. Don't forget, the donor red cell must be washed so that I will make it blank. All the plasma, antibodies, whatever, will be washed off. Okay? I will now mix the donor red cell with the recipient serum and then add my anti-human globulin and see whether there will be agglutination. So if there is an agglutination, meaning that the recipient has antibodies against an unidentified red cell antigen of the donor, that means the blood is not compatible. That's what we do when we are doing cross match. So apart from checking the grouping that is the same group, we also cross match the recipient serum with the donor red cell. So if the person is going to take 4 pints, you must prepare 4 tubes for each of the donor and check individually with the recipient serum. Does that make sense? So that is an indirect Coombs test. What is it used for? I want somebody to tell me what indirect and direct Coombs test. Direct Coombs test is used for to check for antibodies on the red cell of the person, to check whether there is an antibody that is clinically significant on the red cell. On the, what about the indirect Coombs test? Eh? Antibody in the patient serum, and it's used for cross match. So if you want to cross match, you mix the serum of the recipients. So how do you get serum? You are the house officer and you are requesting for grouping and cross matching. What bottle do you use for collection of the blood of the person? You are guessing. Hmm? You use a plain bottle actually. You use a plain bottle, because if you use a plain bottle, it's going to clot, and then the blood will be retracted and you get the serum. So if you are requesting for grouping and cross matching as a house officer, don't collect sample in EDTA or lithium heparin. So you use a plain bottle. So when you send the sample in a plain bottle with the request form to the blood bank, if it gets to the blood bank and you are properly labeled, will centrifuge. By the time the blood sample gets to the blood, it has clotted. So they will remove the serum and go to the blood bank fridge. Go and pick blood to cross match with the serum of the person. Then cut a little bit of it, put it in a test tube, wash with normal saline, wash 3 times. They will now take a drop of the blood from the fridge and mix with a drop of the serum of the patient, then add anti-human globulin, look at it under the microscope. Take 30 minutes to do. Thank you for listening. I hope you have all enjoyed yourself. So somebody, I think that's about all. So you can read this. So somebody should, the person that recorded it should send, to come around and give me the recording.